Hi, everyone. I'd like to thank the organizers for the opportunity to speak. I'm a PhD student at the Computational Biology Department at CMU, and today I'm going to talk about scQuery. So we can imagine a hypothetical situation that might be pretty common is that I'm an experimentalist and I've just profiled some cells, and typically I'll have some first, high-level, surface-level questions that I like to answer about this data, such as what are the cell types that I have, specifically the subtypes. So I might know the high-level tissue type that I've taken the cells from, but I'm interested in a finer grain resolution characterization of those cells. Further, what's that distribution? What does that look like? And also, if I can find that out, then what are some differentially expressed genes between these different subgroups? And the way that you might go about typically answering this is, well, first you'll probably want to do some dimensionality reduction. And if you're just looking at your own data set, then doing something that's unsupervised is pretty common, and that tends to work fine, something like PCA or something maybe specifically for single cell. But when you go to integrate data from many different data sets or studies or labs, and they come from different cell types, that could be problematic. So that just shown here with this toy example of these two different populations, the red and the blue, and they're in 2D, and I want to go to 1D. If I project them onto the principal component, the first principal component, the purple line, they'll completely collapse onto each other, and that makes it really hard to do downstream analysis, and that's because the algorithm didn't have access to these labels. So this could be problematic for large-scale analysis. Another thing that you might do is you might want to look at the expression of marker genes. Marker genes are great when you have them, but particularly for single cell, if they do exist, they're probably few in number, and because of the noise, it's not ideal. So you'd preferably like to do analysis based off of all of the genes rather than just a subset. So what we aim to do with scQuery is to, rather than rely on those things, is to alleviate that by using all of the data that we have for single cell that's out there and enabling some global comparative analysis. Basically, and we need to make this efficient and scalable because of the size of the data. So what you can imagine is that you've profiled your cells and you go to a place where you can upload that, and the system will then, by doing this global comparative analysis, give you a good idea of the cell type distribution that you have and maybe what that looks like. So that's the goal. How do we get there? First, we need data, and we need a lot of it. So we've developed a pipeline to automatically query Geo and Array Express with some frequency to download all the publicly available mice data. And as we're downloading it, we want to label it because, again, I mentioned that we want to maybe do, rather than unsupervised, a supervised dimension reduction. So we can look at the metadata and do some matching to the cell ontology, which is what we use for our set of labels. And what's important here is that you'd want to avoid downloading the author-published normalized data because they might have different gene sets, and pre-processing pipelines are a bit different. So what we do is we download the raw data and we normalize all of it the same uniformly ourselves. And in doing so, the result of this is a very large database. We have over 150,000 cells from over 500 different studies, and we have over 300 different nodes that we've mapped cells to. And what you can then use this for, again, because it's labeled, is when you go to do the dimensional reduction, you can do it in a supervised fashion. And we and others have shown that you can use neural networks to do this, and there were some talks about this yesterday. But the idea is that you have as your input the gene expression, so we have roughly 20,000 genes, and you train the neural network to maybe do something like cell type classification, and then you can take that blue layer, that last hidden layer, as your learned representation, which is of lower dimensions. And for us, that's 100 nodes. So we go from 20,000 to 100. But maybe you've noticed that the big problem with this is the number of parameters here. When you have this fully connected network from 20,000 down to, let's say, 1,000 in the next hidden layer, you already have tens of millions of parameters. So you'd like to constrain that. And one way that you can go about doing that is by incorporating some prior biological knowledge. And Gia in our lab had a paper in NAR on how she did this by using PPI or protein-DNA interaction data. And you can see that those nodes, that basically these modules of genes are much more sparsely connected in the fully connected versions, reducing the parameters. And we have extended this by looking at maybe using the gene ontology, which will give you additional hierarchical information to do this, so we also explore that in this. One other thing that you might care about is that embedding itself. Is it discriminative? Does it help separate? And in that embedding space are similar cell types close together, different cell types further apart. To encode that objective, you can actually use a Siamese or triplet network. There was a talk about this yesterday as well. I won't get into the details of a loss function here, but basically you are, instead of asking the neural network to classify a cell type, you're giving it pairs or triplets of these and asking it what is the distance, the predicted distance between these. And with the loss function, you basically are forcing it to push and pull apart in this embedding space. So the end result of using all of these, we try various different neural networks, we need to evaluate them, and this goes back to this global comparative analysis as our task. We're basically doing an information retrieval task here. We have some new cells, some query cells, and then we have our training cells, and they're both labeled for our tests, and we embed them using the same neural network. So in this reduced dimension, now what we'll do is we'll do an information retrieval, we'll do nearest neighbor search, and the score values that you use usually in this field are mean, average, precision. We have a modification of that to give us credit when we return the wrong cell type, but it's a close cell type. And the point of this figure is basically each row is a different model to do the dimensional reduction, and the columns are different cell types. And you'll notice that in any given cell type, the best performing embedding model is always a neural embedding model. So the rows from top to bottom are these neural networks at the top, the first 3 bands, and then the bottom band are PCA and not reducing the dimensions. And we always perform better with a neural embedding model, but the point of this figure is also to show that it's not perfect, that it's never the same model for different cell types. And we envision that that could be improved as we collect more data, especially for the cell types that we don't have a lot of data for. We can improve in those areas. The way that we make this available to people is through a web server, and you can go and try this. And as a short little demo, you'll go and basically upload your data. And you're first met with a heat map visualization where each row is each of your query cells that you've uploaded, and the columns are cell types in our database. And the coloring is basically how similar your query cell is to those database cell types. And I've selected, for instance, this cluster here, and I actually uploaded brain cells in this example. And so I look and I see, well, as I expected, this is just an example. The best matched cell type in the database is brain. And once you've selected the cluster, you can do further analysis that we make available using this global comparative analysis. So I can look at the distribution of the nearest neighbor, the top hits inside of the database for my query cells, and kind of get a picture of what the distribution looks like. And further, I can also visualize taking the reduced dimension and reducing it further into just 2D, what that looks like for my query cells against some other cell types that we have in our database. And then further, I can maybe look at, within the cell ontology, the hits, the nodes that are most relevant for the query cell types that I have, and explore that. So we've pre-computed some differentially expressed genes that are specific for cell types using our large data set, and that's the left column is the average expression of those. And on the right, those other columns on the right are all some of your query cells, and you can see there's a correlation here. And one thing we wanted to do is to see how well this works for some held-out data. So we did a case study where we looked at recent neurodegeneration data sets. So in this study, they had a CK-P25 mouse model, and the expression of P25, a kinase activator, it is induced, and it results in Alzheimer's-like pathology. And so they had this time series data. Basically, they had 0 from 6 weeks, and they used marker genes heavily in their analysis, which was great. And what we wanted to do is we wanted to see if we could recapitulate some of that using scQuery and not rely on marker genes. And what we found was that when we applied our method, we were looking at basically comparing these different populations of disease and control mice. And we found on the plot on the right, you can see that within the disease population, we were able to see a difference in the classification distribution that scQuery was giving us. And basically, that difference was most significant in some immune cell types like bone marrow cell and macrophage. And what we were also able to see was a increase of this difference as you go from 0 weeks until 6 weeks. That's what the plot on the left is showing, is the p-value, the Fisher exact test p-value between comparing these classification distributions. So we were able to kind of recapitulate the results they found in that original study, and we also did some analysis using the differential expression that we also had, and we found some genes that they found, and we also found some additional ones that were relevant for the immune response that they were finding. So to kind of summarize what we did, we developed a pipeline to experiment with many different neural networks to embed all of this data, and then settled on a few models where we then applied a recent mouse brain case study to see how well they were working, and we made it available as a web tool. But it's definitely not perfect. There are some issues that we're dealing with and that can be improved. So specifically, a big one is handling UMI data. There doesn't seem to be a good standardized way where people are publishing this that we could pull the correct barcodes, and even within one technology company, they might update it. So coming up with a good way of wrangling this data in an automated fashion would give us access to an even larger database, and that's very important. Another problem that we have currently, again, this was related to the first one, is that the distribution of the data in our database is skewed. We have very few examples of some cell types and much more examples of some other ones. And of course, we could always improve the usability of the web server and the performance of those neural nets. And some things that we want to try in the future are, well, these are high-level questions that I was talking about today, but people are more increasingly interested in answering some more specific things like identifying some rare cells in their data, and we feel like we could also help with that with a tool like this, and that we would explore in the future. So with that, I'd like to thank the organizers again, and I'll thank my advisor, Ziv, and Matt is a postdoc who's the co-first author on this work, and Lynn and Ayapa were master students who were very helpful in actually making the web tool that I showed to you today, and I'd also like to thank our funders. So with that, I can take some questions.
Okay.
Thank you for your very interesting talk. So I have a rather technical question, so correct me if I'm wrong there. Based on my understanding, if you use the CMS network, you have an embedding vector in a final output layer, which gives you the dimensionally reduction, is that right?
True, yes.
So my question is that, how would you decide the optimal number of the neurons in your embedding vectors? And so for example, like PCA analysis, you can have PCA1, PCA2,
right,
which basically gives you the most variation on that direction.
Yeah.
And I think this is the second question actually. How do you prioritize the most important dimension from your neural network? Yes.
So yeah, good question. So when we're trying to pick the best neural embedding model, we do a large search over hyperparameters, and one of those is the architectures. So we try different architectures in terms of the number of hidden layers and the sizes of those, particularly the last hidden layer. And so it's a trade-off. So we wanted to achieve some dimensional reduction because of the scale that we're trying to deal with, especially as the size of the data is growing every day. So 100 nodes was a good trade-off we found. We tried like 50 nodes, and the performance wasn't as great. And when we had higher, we still were able to do better with 100 dimensions using the neural embedding model. So that's how we settled on there. It's kind of a little bit arbitrary why we chose exactly 100, but yeah, we searched over a space of different dimensional areas. And when we were comparing to PCA, we always compared with PCA with the same number of components as nodes in our last hidden layers. Does that answer your question?
Yeah, but did you give importance for did you give the importance like a PCA for each of the dimension?
For the neural embedding dimensions? No, we didn't do some sort of, I guess, I'm not sure if I understand, but maybe like assigning an importance to each one of the nodes. We didn't do that analysis in this paper, but there has been some work in GS paper where that was her focus a little bit, was looking at, especially for those sparser architectures, when you're using the gene modules, what some of those modules are representing and how important some of those are for some of the cell types that you're trying to classify.
Okay, thanks. So in your pipeline initially, you had a mapping to your cell ontology. So my question is, can you do without it? Is that a necessary step? I mean, could you learn hierarchies in your embedded space or not? So
it was necessary because of our goal of doing, it was key for the supervised dimensional reduction. So, I mean, just because we want to do that, you need a label set. I'd be worried about trying to learn the hierarchy because of the diversity of the data. I mean, we're coming from all these different studies in different labs. I would be concerned about trying to do that analysis and it just basically learning different labs and different cell type, different labs basically, yeah. So, yeah.
Yeah, I guess somewhat related. Could you comment on how easy you can map the actual published annotations to the set ontology?
Yeah, so Matt was really integral in that, and so the method that we do is basic string matching. It's not anything complicated like more complicated natural language processing models, and that's because, well, the label set is small. The cell ontology doesn't have that many. It's on the order, we use 300 nodes, and the authors usually do a pretty good job of labeling the metadata, but it is noisy, but we've seen that the accuracy is pretty good. I don't have specific numbers on that, but basically downstream, when we do analysis like looking at the differential expression between these groups, the terms that come up make a lot of sense for what we're getting.
So two very quick technical questions. So how does it scale? Is it easy for you to add more data? Do you have to recompute everything? And also, how do you account for the fact that you have all these different platforms from which the data comes with different statistical properties? Is it important? Do you account for that?
Yeah, great question. So for the first one, that's actually something that we are looking at exploring is like when we have new data, do we retrain the entire model with all of that data, or do we somehow use what we've already learned and fine-tune it. We're exploring that, but right now at the scale that we have, it doesn't take all that much time to retrain everything. I mean, it's on the order of hours, not days for now. But that's an interesting question that we're looking at. For the second question you asked, in terms of accounting for the differences in platforms, that's where the labels come in, and we're hoping that the neural network, when we're training it, its supervised information is coming from the cell type label, not from what lab or what technology or what platform. So the hope is that it learns to normalize for that just by virtue of the signal that we're training it on.
But pairs of cells are coming from different experiments, right, from different labs,
yes.
For which you label. Is it a problem that they can come from different platforms?
For the Siamese training, or for, yeah. So the Siamese and triplet training, they're finicky and they have issues, and we have looked into using that, but I haven't been able to find that it improves greatly by making sure that they come from, when we're doing this comparison, they come from independent studies versus just taking different cell types